Hi everyone, my name is Mirami, and today I'll be presenting our project for Dr. Shi's lab on the Enhancer AV toolbox targeting the speculum under the supervision of my mentor Wenhao. Alzheimer's disease is still one of the greatest mysteries in neuroscience. One of the first brain regions to break down is the speculum, which is a major output hub of the hippocampus. This region is crucial for memory and spatial navigation. The challenge is that the speculum is just not one structure. It has different layers and subregions, which has its own function. Traditional tools like transgenic mouse lines are both time-consuming and costly. Maybe they can only target a broad classes of neurons. That makes it very hard to study the precise circuit that fell under Alzheimer's. Therefore, we package this construct to AAVs and then deliver them into the mouse brain. Delivery method including either through the bloodstream using retroorbital injection or directly into the brain using stereotaxic injection. Once expressed, the target neuron glow and let us identify and track the neurons. Unlike traditional mouse models, which takes months to breed, Enhancer AAV can light up very specific neuron types within just a few weeks. That level of accuracy means we're just not seeing a blurry outline of circuit anymore, but we can also zoom in with precision. And because these tools are so adaptable, they open up to possibilities even beyond the hippocampus. Here we can see the result of two Enhancer AAVs, CN3030 and CN3029. The green signal is SYFP2, our fluorescence reporter. The blue is DAPI, a nuclear stain, and the red is GABA, which marks inhibitory neurons. Looking at this panel, we can see that both viruses have specifically target excitatory neurons in the speculum. Since the green SYFP2 overlap with the excitatory cells, but not with the GABA inhibitory cells, the key takeaway here is that both enhancers have achieved a strong specificity, and then with CN3030 showing a specifically robust labeling on the top, which are called the deep layer of the speculum. To make this more rigorous, we manually counted the SYFP2 positive neurons. This graph shows excitatory specificity for both enhancer AAVs across the injection methods. We can see that both viruses have achieved almost 100% specificity for excitatory neurons in the dorsal speculum. That confirms that they are not only precise in labeling, but they can also consist in different delivery methods. In summary, we've shown that Enhancer AAV have selectively labeled excitatory neuron in the speculum with a high accuracy. And therefore, our next step is to quantify the other layer-specific labelings, compare such as superficial layer with deep layers. Test Enhancer AAV in the pre-speculum, which contains the head direction cells, the brain's internal compass. And even more, we can investigate why Enhancer AAV cores sometimes give slightly different labeling patterns, which may reveal new biology about circuit diversity. Thank you for listening.